Thank you all for coming. Thank you for inviting me. It's really, it's been a great meeting so far. So I am going to talk about cortical circuits underlying auditory decisions. And in spite of what Moshe says, I could also give a fun talk on connectomics, but in deference to him, I will not. Okay, so right, so my lab is interested in circuits, and I think it's important when thinking about the relationship between circuits and behavior to go back to the really early work that has been done on this subject. This is the earliest work that I'm familiar with. This is the compelling story of Descartes, who 300 plus years ago was interested in the circuit by which we withdraw our foot from a fire. And he proposed a specific mechanism for that. And the mechanism he proposed was that the flames of the fire displace the skin, which pulls on a tiny thread. The thread opens a pore in the ventricle, which is conveniently labeled F up there. The animal spirits flow from the ventricle through a hollow tube, and then they inflate the muscle of the leg, causing withdrawal. So I'd like to build on, I would say that, admittedly, we would revise that model somewhat. We know that the animal spirits flow from a point out here. But really what I find compelling about that particular explanation is that it's very mechanistic. And to my knowledge, it really is the first really mechanistic description of a behavior, in that one could imagine taking inspiration from this and building a reflex, building a machine that actually performs the task that he set out to describe. So what is missing from here is, because it's a reflex, is any notion of taking inputs and processing them before producing the output. Here there's a stereotyped, very simple stereotyped relationship between the inputs and the output. So when I started my lab, we started focusing on simple auditory decisions, and this is an example of the simplest decision that we could come up with. Here we have two sounds, rotate. So this was my younger son playing with a Rubik's Cube before he'd really figured out how to do it on its own. We had to coach him. And so we would say rotate left, and he'd rotate, and then rotate right, and he'd rotate, and thereby he would quickly solve it. And what this reflects is an arbitrary association between two inputs, two sounds, and two outputs, two actions. So this model system has some limitations. And so in view of those limitations, when I started my lab, we actually turned to this model system, the rat, and more recently the mouse. And we can do the same thing. We can train rats to listen to sounds and perform actions. And so here is the basic paradigm that we use in the lab. This was developed actually when I was next door to Zach Mainin, and the original paradigm was worked out. Zach and I started our lab side by side with the goal of getting rats to perform the kinds of behaviors that before then people had only trained macaques to perform, really. So the idea was to get the rodents under tight behavioral control so we could apply the kinds of approaches that people had used in studying neural correlates of processing in macaques. So here is an example of a rat in action. We're looking from above at a 3-port chamber. He is trained to stick his nose into the center port. This elicits a stimulus, and then he goes left or right depending on what the stimulus is, and if he goes to the appropriate port, he gets a little drop of water. And because he is water deprived, he's highly motivated to do this for many, many trials. So here's just an example. And what I hope you get out of this is that he really sort of understands what he's supposed to do. Each of those trials is correct. He'll keep doing this for dozens and actually up to maybe 1,000 trials a day. Okay, so why are we using rodents? Well, just very briefly, we can train dozens of subjects in parallel, and each of these animals for a simple task like this takes under a month to train. And of course, the great payoff of using rodents is experimental accessibility, so that nowadays with the various optogenetic and calcium type imaging reagents, we can monitor and manipulate the circuit at a level of resolution that really I think I wouldn't have predicted 10 years ago when we started this. Okay, so I'm going to focus on 2 questions for today's talk. The first question is, how does information get from the ear to the muscle? What are the neural pathways by which information gets from the ear and out the muscle to drive behavior? Okay, so we're pretty confident, based on work from Ellie and others, that sound comes in through the ear. And eventually, he showed you the circuit by which it gets to the auditory cortex. And we're pretty confident, again, that it is using muscles that the animal moves, and they cause the motor responses. But somewhere around here, we lose track of what happened to the information. Okay, so traditionally, we know that the information gets up to the auditory cortex, and we know that auditory cortex projects to many structures, of which I've listed a few here. And when we started the study, we really didn't know where to look to think about how the information propagates beyond the auditory cortex to affect behavior. And out of all these choices, I'm going to argue that the one that is important, or at least one that is very important in this task, is the auditory striatum. So that the key projection, or a key projection in this pathway, is the projection from the auditory cortex to a part of the striatum that receives input from auditory cortex and auditory thalamus. And so the second question is, what is the neural substrate underlying the acquisition of the tone direction association? Okay, so we take a naive animal, we put it in there. It doesn't know to go left for high sounds and right for low sounds. We train it for a while, and eventually it behaves like I showed you. Something in its brain has changed to instantiate the association between the tone and the direction. Presumably, that thing that has changed is the strengths of some synapses somewhere in the brain. But specifically, what I'm going to argue is that the synapses that change during learning, during the acquisition of this task, are the synapses that connect the auditory neurons in the auditory cortex to neurons in the auditory striatum. And they change in a very specific pattern in a way that we think can explain what's going on in this task. Okay, so those are the 2 questions I'm going to set out to answer. In the first part, I'm going to focus on how activity in the auditory cortex to the striatum mediates decisions, and then later, in the second part, plasticity. Okay, so this first part is really the work of a very talented former graduate student in my lab, Peter Zemensky, who is now doing a postdoc with Tom Mersick-Flogel. And really, he was the one who drove this project. Okay, so Ellie already reviewed the basic organization of the auditory system. The key thing from our point of view is that the sound, as soon as it comes in, undergoes a spectral analysis. So it's there are neurons, low-frequency neurons and high-frequency neurons in the cochlea, and that the organization in terms of low to high frequency is preserved up to and including the auditory cortex. So within the auditory cortex, there is a map of frequency space so that one side of the auditory cortex the responses are predominant, or the neurons are predominantly driven by lower frequency sounds, and on the other side by high frequency sounds, okay? That's the key fact that we're going to exploit for this to understand how the information propagates. Okay, so the stimuli that we use for this study are a variant of the tone cloud stimuli that Ellie introduced, but they're a special variant in which stimuli are either pure low frequency, pure high frequency, or a mix of low and high. Okay, so each one of these little, these are spectrograms. This is frequency and time. You can think of these as musical notes. So this is a bunch of relatively low frequency sounds, and I'll just play them quickly. I'm not sure if you can hear that. I think usually the high frequency one is a little bit easier. Okay, so they basically sound like the transporter from Star Trek, and then the middle frequency sounds are here. Okay, and so the animal is asked to go right when there are more high frequency than low frequency tone pips, left when there are more low than high, and in the intermediate ones, he's asked to judge the relative number. And when there's a perfect balance, he can't possibly get it right, so we reward him randomly. So the sort of organization of this stimulus is actually motivated by the classic random dot stimulus developed by Bill Newsom and colleagues to study decisions in MT of the monkey. So this just shows that the rats can learn the task and that we really have them under behavioral control. At the extremes, the rats perform essentially perfectly with essentially no errors. And in between, they make some errors, and the errors sort of fall off in the way that you'd expect from them making some kinds of mistakes in judging the relative numbers of low and high frequency sounds. So the simple sort of model that we had going into this is that there is a population code in the auditory cortex whereby each sound activates some neurons, and somehow, somewhere in the brain, there's a comparator that compares the number of high and low frequency neurons that are activated, and if there are more high than low frequency neurons, the animal goes left, and otherwise it goes right. Okay? So in light of that simple model, we predicted that if we could activate a few additional neurons, shown here, if we could cause a few extra neurons to fire, we would shift the animal's, we would affect the animal's perception. We would cause it, if you like, to hallucinate extra tones and thereby shift the psychometric curve. And if the animal were merely hallucinating a few extra tone pips every time we stimulated the neurons, this would cause a shift in the psychometric curve of this form. And if we fit the psychometric curve, we could extract the amount of the shift by this bias term. Okay? And so I'm going to be showing you the effect of these kinds of stimulation experiments on the bias and also the slope of these psychometric curves. Okay, so this was, we started these experiments in the relatively early days of optogenetics, and so we were still sort of fooling around with how best to deliver channel rhodopsin and light. And although we were really interested in targeting a specific pathway, the pathway of the pathway that takes the signal out of the auditory cortex, we started with what we thought would be the simplest experiment, where we simply stimulate neurons in the auditory cortex sort of randomly, ubiquitously independent of where they carry their information. So we used a virus to introduce channel rhodopsin into the auditory cortex, and then we had fibers in the auditory cortex. The fibers were bundled with tetrodes so that we could stimulate a little population of neurons in the vicinity of each of these tetrodes and also record the activity, the tuning properties of the neurons in the vicinity of the electrode. And so we could correlate the tuning properties of the neurons that we were stimulating with the animal's response, the behavioral response to stimulation. Okay, so these experiments were very disappointing. As you can see, Peter tried 10 rats, so he really tried to get this to work. And there is absolutely no orderly relationship between the tuning at the site that we were stimulating and the effect, the bias, the shift in the psychometric curve. Now, that's not to say that there was no effective stimulation. These points, this is bias. These points out here, many of them, are highly significant. The animal was really definitely being affected by the stimulation. It's just that we could not predict the effect of the stimulation by knowing how the neurons in the vicinity of the electrode were tuned. What we did see was a pretty consistent decrease in the animal's performance. So basically, what we got from these experiments was that if you stimulate neurons in the auditory cortex sort of randomly, you screw up responses in the auditory cortex. You screw up the animal's perception of the sound. So not the sort of subtle manipulation that we had in mind, more like a hammer. Peter was sad when he got these results because these took a long time and his thesis was on the line. There are many possible explanations for why these experiments failed. We still don't know. My favorite explanation is that the way we delivered the virus, we were stimulating both excitatory and inhibitory neurons, and perhaps sometimes we were preferentially affecting the inhibitory neurons. There are some other possibilities. Rather than dig down and figure out why these experiments failed, we instead pressed on and used a different strategy, which is one that would allow us to target a particular pathway out of the auditory cortex. So I already gave away the answer, which is that, that the relevant pathway is the striatum, but I will say that that happened, that was really Peter's decision to focus on the striatum. My lab had no knowledge of or expertise in the striatum, and when he started these experiments, he came to me and said, so what do you think about the striatum for a pathway for us to target? And I said, no, you should focus on the sort of pathways analogous to the ones that have been identified in the monkey, perhaps the pathway from auditory cortex to posterior parietal cortex, or the rodent analog of the frontal eye fields. And he said, and then came back a week later and said, what do you think about the striatum? I said, no. And after 3 or 4 of those conversations, he stopped asking me and just ignored my advice, and then went to the striatal neurons. And of course, that turned out to be a great choice. So why did he do that? Well, in retrospect, it was a really good choice, and both because what we know about the striatum suggests that it would very likely be involved in this kind of highly practiced task, as we've trained these rats to perform, but also because there's a great strategy in looking where the light is brightest. And the answer is that there's a very strong projection from the auditory cortex to the auditory striatum. So if we had chosen to pursue one of these other pathways, we would have been looking at a relatively small number of fibers, whereas the pathway from auditory cortex to auditory striatum is one of the 2 or 3 strongest pathways out of the auditory cortex. And that's shown here from this slide from the Allen Institute. And so the part, I should say that the part of the striatum that we're looking at really is exclusively, it really should be described as the auditory striatum, because it receives inputs only from auditory cortex and auditory thalamus, okay, not from other parts of the cortex. So therefore, it really seems focused on processing something having to do with auditory information. And a lot of what you might have heard about the striatum comes from other parts of the striatum that receive inputs from other parts of the cortex. Okay, so how do we target these neurons? Well, we developed a strategy based on using a retrograde virus. So we had previously been working with a retrograde herpes simplex virus that, when we inject it into this area, goes backwards and then infects the neurons that project to that area. In this case, we used a retrograde Cree that I believe we got from Andreas Fluti's lab, so if he's somewhere, thank you from that. And then we also injected a adeno-associated virus in which the expression of channelrhodopsin depends on the expression of Cre. So only the neurons in the auditory cortex that project to the striatum are going to express channelrhodopsin. And this is just a picture of those neurons as expected. They're predominantly layer 5 neurons with the occasional layer 2-3 neuron mixed in. So once again, we can look at the strategy. So here are neurons in the auditory cortex that project to the striatum. Now it turns out, and we'll come back to this, that the projections in the auditory cortex are topographic, so that high-frequency neurons in the auditory cortex project to a high-frequency region of the striatum, and similarly low-frequency cortex to striatum. So the idea is that only the neurons in the auditory cortex that project to the striatum are infected. So if we put in a light here to stimulate, then we're preferentially, or we're selectively stimulating this pathway. And voila, that experiment actually worked. So we now see an orderly relationship between the preferred frequency at the point that we're stimulating and the choice bias, okay? So this is the sort of, really the punchline of the first part of the talk, this relationship between where we stimulate and the bias, suggesting that the neurons that are in the high frequency part, when stimulated, bias the animal to produce a high frequency, bias the animal as though it heard extra high frequency sounds. So Peter was very happy about that. We also did this experiment in a different way, which I'm not going to show you, where we directly stimulated the terminals in the striatum, exploiting the tonotopy in the striatum. And we did some other control experiments. But the set of experiments I'm going to show you now, there is another set of experiments I'm going to show you now that I think are interesting, showing because these experiments showed only that specific corticostriatal activation can affect auditory decisions. And a caveat of this and pretty much any kind of microstimulation experiment is that this does not show that these neurons are normally involved in the behavior, okay? The fact that you stimulate a population of neurons and that produces some effect doesn't show that stimulation of those is normally involved. If I hit your knee and your leg moves like that, that doesn't imply that normally when you're moving like that, it is due to somebody hitting your knee, right? And that's just illustrated here. So there are really two models of what could be going on here. There's the co-pilot model, according to which these neurons are normally involved in affecting the decision. But there's also the backseat driver model, where the backseat driver is most of the time, one hopes, silent. But then if suddenly the backseat driver says, turn right, you will find yourself turning right, even without knowing what you're doing. So the way to distinguish these experimentally is that if you silence the putative backseat driver, you should see no effect on the behavior, right? Because the contribution under most circumstances of the backseat driver is minimal. But if these neurons are normally involved, then silencing them should have an effect. And in fact, the effect of the silencing should be opposite the direction of the activation, right? So you should see an anti-bias. So activating a backseat driver can affect decisions, but silencing him or her will not unless he or she is the co-pilot. So we use the exact same strategy as before. Retrograde Cre in auditory striatum, fibers, tetrodes in auditory cortex, except now instead of putting in channelrhodopsin, we put in arch, which when activated by light shuts down neural activity. And voila, here's the result of those experiments. There is now an orderly relationship between the preferred frequency of the area of the auditory cortex in which we're silencing and the bias, but now the slope is in the opposite direction. So this reflects an anti-bias of the animal, as though we're taking away tone pips when we silence these neurons, okay? So from this, we conclude that corticostriatal neurons normally participate in these decisions. They act as pilots in these behaviors, not backseat drivers. Okay, so now I'm going to turn to, so basically, from that, what we conclude, and I should actually be very clear about what we can conclude from those experiments. We targeted a population of neurons defined by the fact that they project to the auditory striatum. But because of the way we did the experiment, we cannot rule out the possibility that it is some other connection of these neurons that is actually important. Okay, so these neurons project not only to auditory striatum, but some of them project to other sites. Because of the way we did the experiments, we can't rule out the possibility that it is one of those other projections that's crucial. We don't believe that to be the case, in part because of some of these experiments, but at the time we did those experiments, there were no reagents that would allow us to distinguish those possibilities. So that's a little asterisk by this result, that it is the neurons that project to the auditory striatum that are involved in this decision, but not necessarily, based on these experiments, the striatal connection. So now I'm going to focus on the second part, which is that plasticity, auditory cortical striatal plasticity mediates learning this task. So the strengthening of the synapses that from the auditory cortex neurons that terminate in the striatum, or that there is potentiation of synapses from the auditory cortex to the striatum that is invoked by this task. And I'm going to show it to you. I'm going to show you two sets of experiments. The first set of experiments are in vivo, where we monitored the strength of the synapses during the course of learning. And the second set of experiments are in vitro, where we take animals in which we've already trained them, make slices from their brains, and then monitor the strength of synapses across the striatal gradient. Okay, so this is joint work between Peter and a very talented postdoc, Xiaoji Zhang, who is starting a faculty position at Stony Brook next month, actually. Okay, so now I'm going to present you a model of what we think is going on. This is just a provisional model. You can think of it really as a way to make sense of the observations that I'm going to show you. I don't want to take the model overly seriously. It's certainly not the way I would have designed a brain, but it's the simplest model that we could come up with that can account for the observations that we have. So the model is based on a couple of relatively indisputable facts. So there are two sides of the brain, a left and a right. There are two auditory cortices, two auditory striata. And there are, as I'll show you in more detail, the projections from the cortex to the striatum are topographic. And they're largely, but not exclusively, ipsilateral. So the last little bit of information is that when you stimulate the striatum during this task, you tend to bias animals to make a contralateral motion, okay, a contralateral decision. So if you stimulate the left striatum, you will tend to promote right choices, and if you stimulate the right striatum, you'll tend to promote left choices. So the main sort of conclusion from our study is that what you see is a preferential strengthening in trained animals of low-frequency synapses on the left auditory cortex to the left auditory striatum in animals trained to associate low with right choices, and similarly, a high to high on the other side, promoting a left choice. So if you play a low sound, that will promote a right choice, and if you play a high sound, that will promote a left choice. In animals that have been trained the other way, the pattern is the opposite. Okay, so keep this model in mind as I show you some observations. Okay, so we developed a method to monitor synaptic strength in vivo in animals learning to perform this task. Okay, so this is synaptic strength of the corticostriatal pathway during the course of acquisition of the association. And to do this, what we did was we put channelrhodopsin into the auditory cortex, and then we put our light and stimulating fiber in the auditory striatum. And in that way, we could elicit action potentials in the neurons from the auditory cortex that project to the striatum. And when we did that, we saw, responses that look like this. Okay, so this is 5 milliseconds. And as a former hippocampal slice physiologist, this looked very familiar to me, because it looks essentially like what you get when you stimulate the Schaffer collaterals and record in CA1 of the hippocampus. And so if this had been a hippocampal local field potential, the way we would interpret it is that this portion would be, the slope of this little portion right here would be proportional to the synaptic strength. So we did some pharmacology, and indeed it seemed consistent with that interpretation. So when we put in blockers of inhibitory transmission, picrotoxin, that had no effect on this. But when we put in blockers of excitatory transmission, we saw that this component here completely went away, whereas this component here was unaffected. And then when we put in TTX, which blocks all axonal transmission, there was still this component, and a little extra component was removed. And so we interpret this, and I'll provide more evidence for this interpretation a little bit later, but we interpret the slope here as proportional to synaptic strength. And I should just say that in terms of plausibility, so in general, you can't stick an electrode somewhere in the brain, elicit a response like this, and then interpret the early component as proportional to synaptic strength. But the reason that that works in the hippocampus is that the hippocampus is one of the few structures in which there is no recurrent excitation. So the CA1 region of the hippocampus has no recurrent excitation. The CA1 neurons receive input from CA3. They excite local inhibitory neurons, but they don't excite each other. And that's why you can, that's basically why people study synaptic transmission in the hippocampus. So it turns out that the striatum is another such structure, something I had not appreciated, because the vast majority of the neurons there are inhibitory, the medium spiny neurons, and of the remaining, that's 95% of the neurons, and of the remaining 5%, none are glutamatergic. So the only source of glutamatergic input to the striatum comes from the cortex and the thalamus, and we were specifically targeting the cortical inputs. So that provided sort of a plausibility argument, and this pharmacology and some later pharmacology, I think, really supports the idea that we can interpret this as synaptic strength. Okay, so learning this task strengthens cortical striatal synapses. So the way we did the experiment is first we tested the strength of the, we put a fiber in, by we I mean Xiaoji. She put a fiber into the auditory striatum in an animal that had been injected in the auditory cortex with chanorodopsin, and then stimulated, and that produced a response. So this is a naive, untrained animal, did that for a few days, got a baseline, and as you can see, the baseline is surprisingly, at least to me, surprisingly stable. So this is the average of responses for each of 4 days in which she did that experiment prior to training. Then on the 5th day, she trained the animal, repeated this, and saw that that response had gotten larger. And then she did that a few more days, and you see that it got larger still. And that's summarized here, where there's no performance for the first 4 days for the baseline because it hasn't started training the animal. But as soon as the performance starts going up, you see that the slope goes up as well. All right? So now you see that there's a correlation. You see basically that the strength of this input tracks the performance. And that was true just as predicted by the model. In fact, that's what motivated the model. It was true in cases where the site of stimulation promoted a contralateral choice. In sites where it did not promote a contralateral choice, rather an ipsilateral choice, we saw no change in the synaptic strength, okay? And those data are summarized here, where the size of the slope change, the magnitude of the slope change, is proportional to the frequency at the site of the stimulation. Okay, yeah, this is just a control experiment showing that it doesn't happen when you train the animal on a visual task, for example, as predicted. Visual training produces no change. But then when we take that same animal and train it on the auditory task, the synapses get stronger. Okay, so that's in vivo evidence during the course of learning that this particular population of synapses is strengthened while the animal learns the task. In vitro, we also found that learning shapes the synaptic strength across the tonotopic gradient in the slice. So back to this picture, this model predicts that if we can make a slice of the striatum in which we can compare the inputs to the high frequency and the inputs to the low frequency part of the striatum, we should see that depending on which side of the striatum we're at and how the animal is trained, we should see a gradient in this synaptic strength, okay? And so here we exploit the fact that there is a topographic projection from the auditory cortex to the auditory striatum. So we make 2 injections in the auditory cortex of different colored viruses, look at their termination in the auditory striatum, and you see that there's a red zone, a green zone, and a transition zone, indicating this sort of orderly projection to the striatum. And so that leads to this experimental design. Shaoji found a way to cut slices that preserves the tonotopic axis very nicely. The green fibers are green because they're expressing channel rhodopsin. And then she stimulated different portions, different spots within this slice and recorded the local field potential at that particular point in the slice. And the prediction is that one side should be, the synaptic strength on one side should be greater than the synaptic strength on the other. And so here we were able to do a more careful dissection of the synaptic, of this synaptic input to the auditory striatum. So here's the control. So this is a recording at a particular spot in the slice. Here's a little spot of pulse of blue light. And then, let's see. So you can't even see that, but there was no change. I don't even remember what the... so then here's a GABA receptor blocker. Once we put in blockers of glutamatergic transmission, this big component goes away. If we block all synaptic release, that has no extra effect. And then TTX blocks this additional component. So basically what we conclude is that this big thing here is proportional to synaptic strength. This thing is probably mostly an axon volley. And then there's an additional component that has to do with the channel rhodopsin current. And here, this was an experiment that one of the reviewers had us do, but I'm particularly pleased with. So even though there is at least 30, 40 years of hippocampal physiology in which the local field potential is used as a measure of synaptic strength, I was unable to find a single example where somebody explicitly showed that the synaptic strength that you get by measuring the local field potential extracellularly is linearly related to the gold standard, which is measuring it intracellularly. And so we actually did that experiment in this system and found a perfect linear relationship between synaptic strength measured by the gold standard during an intracellular recording and the simultaneously measured synaptic strength as measured by the slope of the local field potential. So this really, to my mind, lends support to the idea. Actually, I think this confirms that this model can be, this measure can be used to, as a measure of synaptic strength. Okay, so here's the result. In one slice, she recorded a dozen or so spots and found a gradient in the synaptic strength across the a slice. And when she did that in 7 animals trained one way, she found that the gradient went one way, and 7 animals trained the other way, the gradient went the other way. So this really shows that training an animal to perform this task sets up a gradient of synaptic strength from the auditory cortex to the auditory striatum. And just the effect was so strong, we actually analyzed it slice by slice, animal by animal. And it turns out that, so the question is, if you take an animal, train it, make a slice, make this measurement, and then ask, can we tell whether the animal is trained to associate high tones with a left choice or high tones with a right choice? The answer is, yes, you can. You can tell with 100% accuracy in 14 out of 14 animals whether the animal had been trained to associate low with left or low with right, just by looking at the slope. So in other words, you can read the mind of a dead animal. Of course, when we did the press release, we got a lot of questions about whether this could be extended to, figuring out where grandma buried the treasure. I said we weren't quite ready for that, but because I don't want to oversell the result yet. Okay, so that's basically it. In the remaining minute or two, I'm going to just wrap up and point out that although we studied the association of high or low frequency sounds with left and right choices, we think that this generalizes to arbitrary stimuli, except that we can't, at this point, test that yet. But we think that if there were one pattern of activation in the auditory cortex that we train the animal to associate with a left choice, and another pattern that we train the animal to associate with a right choice, that those two patterns would establish the appropriate set of connections in the auditory striatum, and that the mechanism would be exactly the same. The problem is that right now we only have very crude measures that allow us to read out the activity of large populations of neurons. But we would like to be able to get this to the level of single neurons and thereby test whether it generalizes in this way. And toward that goal, we're developing a head-fixed two-photon imaging, head-fixed behavioral preparation for two-photon imaging. And basically, I was in the last year, we've managed to take the exact paradigm that my lab and quite a few other labs by now have been using to this sort of two alternative choice behavior and move it to head-fixed mice. So we, instead of having the animal move its entire body, we train the animal to merely lick one of three ports. So now the animal initiates. So this paradigm is formally equivalent to the other one, except that the readout is slightly, the actions are slightly different. So the animal is trained to lick a center port to initiate the trial, and then lick right or left to indicate its response. And you can see that we can get very nice behavior, pretty much as nice as what we have in the freely moving rat. So ask me later if I think mice are, well, I'll tell you now that you can ask me again, but I'll give you the same response, which is I don't believe that mice are any dumber than rats, at least with respect to the kinds of tasks that we train animals to perform in the lab. And that I think really it just reflects that we're not as good at training mice yet as we are at training rats. But every time we've tried to train mice to perform a task, we found that they can perform just as well as the rats, basically. And just to, last slide, this is the head-fixed. So we're looking at a mouse, a head-fixed mouse. Here are the three ports. And let's see. It's actually running on a ball, but that's just to keep it happy. The business end of the mouse is its tongue. So it initiates the trial. And that was the very high frequency thing you heard was the low frequency. The high frequency sound is actually ultrasonic, so we can't hear it. But you can see that he's happily licking away. Looks up, maybe not so happily, and just from that picture. There we go. Anyway, so to summarize the second part of the talk and the end of the talk, auditory corticostriatal plasticity mediates learning in this task in vivo and in vitro. And the third part of the talk, which I won't be giving right now, is just sequencing the connectome. We're developing a method to determine the wiring diagram of an entire mouse brain for $1,000 in a few days, but the margin is too small to. Okay, so I'm going to finish up with my just thank the acknowledgments, my collaboratome. Really for these experiments, it was really Peter and Xiaoji who did all the work, and my fundosome. We got support from the Paul Allen Family Foundation, the Swartz Foundation, NIH, and Cold Spring Harbor. And thank you for your attention.